All right, let's reconvene, get to the rest of our indicators, and then data interpretation. So the next one we're going to talk about, we finished aquatic invertebrates, we're going to talk about algal cover on the stream bed. So algae are, pigmented, single or multi-celled life forms that produce their own energy through photosynthesis. For our purposes, we are including cyanobacteria, we're including diatoms, and we're including soft-bodied algae. So no plants, no bryophytes, just cyanobacteria, diatoms, and soft-bodied algae. The indicator is used in both snow and non-snow influence regions. And we also count live or dead mats. And dead mats can be particularly important in a dry reach. Live mats, you tend to have bright green or dull brown colors. If you have a lot of diatoms, they tend to be golden brown sheens, kind of slippery and mucus-like. And when you're talking about dried mats, they're often brown or powdery white. If they're fresh, they might have a greenish hue. So like I mentioned, we have a variety of different algal forms that we count in this indicator: green, red, blue-green, diatoms. They're all good. We do not count lichens. We do not count iron oxidizing bacteria or other kind of microbial films, and we don't count mosses. Like I mentioned, we count both dead and dying algae, just like we do on living algae. This photo shows dying algae on the left, this sort of brown color, and then the desiccated dry algae is this powdery white stuff on the right bank. So these dead or dying algae, it's often bleached looking, these sort of powdery white deposits. It can be either just located in a few pools that have dried out, or it can cover a huge extent of the reach. Do be careful to not confuse salt deposits for dried algae. They'll look very different under magnification, or even just a close squinting even can be enough. But just make sure you're not looking at salt deposits. You want to estimate the entire cover over the stream bed in the primary flow channel. If you're in a multi-threaded system, we have in the user manual a sort of a guide to what different levels of cover look like, whether it's under a dispersed or a clustered distribution. And we have some photos that go along with each. So here we have what low cover looks like. It's probably zero cover on the right and close to zero on the left. And high cover on the left is what it looks like in flowing conditions. We have large Cladophora mats, and here's what a dried stream looks like on the right, and that's well over 10%, probably actually over 40 in both cases. When you're looking at algal mats, you want to make sure you're able to distinguish between local growth versus deposits of upstream algae. And the signs are usually pretty obvious when you see them. It's going to be like snagged on a boulder or up in a tree branch. It'll be above the high water mark. So if you see your algae that looks like it deposited and it hasn't subsequently started to grow locally, then you want to flag that as separate. But if you have local growth or you have a combination, then that's fine. Those are treated the same. You record this on the field form. This is basically identical to what we have in the Arid West, but we have slightly more refined categories in terms of cover. And so using those diagrams in the user manual can help you distinguish between these different levels. The Arid West used fewer categories. Fish abundance, that's the next indicator. So fish, we don't actually include any sort of directed sampling targeting fish as part of this method. There's no electrofishing or seining. You're simply recording the amount of fish you observe while conducting your assessment and measuring other indicators. Polarized lenses can be great for observing fish. If you have a lot of glare and reflection, the polarized lens will really cut through and help you see what's going on below the surface. And you're best assessing this during your initial walk of the reach. If you see dead fish, they don't count. You can note these on the field forms, but they're not counted towards the abundance. You're not really sure where that dead fish came from. They can be transported by wildlife quite easily. We don't require that you identify any fish with the exception of non-native mosquito fish. Otherwise, it doesn't matter if the fish you're observing are native or non-native. We score the abundance of fish on a qualitative scale ranging from 0 points to 3 points. 0 means that you don't observe anything. 3 means that they're easily and consistently observed throughout the reach. And then we have some sort of we have weak and moderate for intermediate levels of abundance. And beyond that, we also allow half score. So you can put in a 1.5 or a 2.5 if you feel like that is appropriate for the reach you're assessing. The Arid West method did look at fish, but it didn't quantify the abundance. It was simply presence or absence. I mentioned that mosquito fish don't count. There are no native mosquito fish within the Western Mountains, but they are native to small portions of nearby portions of New Mexico and Texas that abut the Western Mountains. If you note them, if you see them, you can note them, but they're not used as an indicator of fish abundance in the Western Mountains. Some of the more common fish in the Western Mountains, as determined by the National Rivers and Streams Assessment, sorry, this actually is for the Western Mountains, not the Zurich West, rainbow trout, mountain whitefish, brown trout, long-nosed dace, brook trout. So if you want to learn to like key in these species, that would probably be helpful. In the beta S-dam for the Western Mountains, fish are both a regular indicator and a single indicator. A single indicator can change a classification of ephemeral to one of, quote, at least intermittent. Within non-snow dominated regions, abundance alone is used as an indicator, but across both regions, it's a single indicator. Single indicators are not an off-ramp to stop collecting data. You should still finish the protocol. This may help you get more precise classifications. So it's not just at least intermittent, but specifically perennial or intermittent. And it can also provide other information that is helpful that is normally collected as part of the assessment. When you record these on the field form, you simply record your score from 0 to 3. In this area, photos are recommended if you're able to capture any. And again, the biggest differences are that the Arid West uses fish as a single indicator, and the Arid West uses only presence, not abundance. The last biological indicator is differences in vegetation between the channel and surrounding uplands. Okay, so in general, when reaches have long duration flows, you have vegetation species of plants that dominate riparian zones that are really well adapted to those flowing conditions. And these same species are typically absent or rare from adjacent uplands. If you have species that are found in both locations, they typically grow more vigorously near the channel than they do in the adjacent uplands. This indicator is used in the non-snow influenced regions, but it can be used as supplemental information across the entire Western Mountains. This is another sort of a visually estimated score where you're giving them a rank from 0 to 3 on an ordinal scale based on your visual estimation. And again, half scores are allowed. And I'm going to walk through some examples of low and high scoring reaches. This is, in the photo, a poor reach. There's not a strong differentiation between upland plants and plants growing in the channel. There's no compositional differences. There's no density differences. So it's the same plants growing at the same density at the channel and above the channel. This one is weak, and you can argue whether it's a 1 or a 0.5 or a 1.5, but here you have clearly more vigorous growth within the channel. You can't actually see the channel because the growth is so vigorous, whereas the uplands are surrounded by grass and herbaceous plants. So there's clearly a difference, but none of these plants are really adapted to wetland conditions here. And some of these shrubs are growing in uplands. So there aren't strong compositional differences. This is an example of a moderate channel. You have a distinctive riparian corridor with some plants that are growing, at the margins that aren't found in the uplands. But there's plenty of terrestrial vegetation that does actually approach the channel itself. So there's a good deal of compositional overlap, but there is some distinctiveness. And then strong, you have dramatic differences between the stream corridor and the adjacent uplands. You have a lot of adapted wetland plants or riparian plants growing along the reach, whereas here the uplands are dominated by pine. So it's a pretty strong and striking difference here. So factors that can raise your score, you have a lot of, like I said, wetland indicator plants based on the National Wetland Plant List. You're running through a meadow is another good example. You're not comparing the plants at the channel to just further within the same meadow. You're looking at the uplands around the meadow. Scores will be lower if you have many upland or terrestrial species near the channel, and there's really not, or there isn't much riparian vegetation to speak of at all. This is, maybe common in more altered settings where, most of the vegetation is managed and removed. You can estimate this indicator in the desktop, but whenever you do that, you should make sure to verify in the field. Desktop estimation is going to be feasible in some areas, like the two photos at the top show, a distinct riparian corridor and the lack of a distinct riparian corridor in two different examples. But it can also be really hard to see as in the aerial imagery, depending on the setting, and that's sort of shown in those two lower photographs. The reason why you want to verify in the field, two reasons. One, if you can't see it in the field, in the aerial imagery, you have to look in the field. But also conditions might have changed from that photo, and you want to really base your estimate on current conditions. There are situations where this indicator is particularly difficult to measure. This is a photo of a stream in the Sierra Nevada of California that, underwent a pretty severe wildfire, and all the vegetation has basically been killed in this fire. Other situations are, heavily urban areas where it's just, there's development goes right up to the channel, or areas under heavy snow cover. So obviously, if there's snow cover, come back after the snow is melted. If you have wildfire, come back after the plants have started to recover from the wildfire. You can try to look for early sort of colonizers that are adapted and see, is it, wetland riparian plants growing near the channel, or is it upland plants? If necessary, you can make a provisional assessment based on conditions that are, historic conditions from before the fire, or based on nearby, like, maybe just outside of your project area, you can see the same channel with more or less consistent conditions, but it's more easy to observe this indicator there. If you do make these kinds of provisional assessments, make sure to explain that on your field forms. So here again, you just record the number from 0 to 3. Half scores are okay. When scoring this indicator, we recommend two photos, one showing vegetation near the channel, one showing vegetation in surrounding uplands. It's not often easy to get a single photo that captures this indicator from one perspective. So occasionally you can do this, but two photos is usually the best way to do that. This one is not used in the arid west, but a related indicator, the number of hydrophytes growing in or near the channel, that was a prominent feature of that method.